All right, are there any questions?
Can you say the next generation is a tutorial in the center?
Yes. Yeah. There is a next generation tutorial at the center, all right?
Who is going to present?
Her name is Nushin Ghaffari, all right? She's going to do, I think you guys know about it, right? I mean, yeah. So it's from 3:30 to 5:30 today and tomorrow. Okay.
Oh,
And I think there are already 25 people signed up because I saw a lot of chairs in the.
I didn't know that. Can I sign right now?
You can show up. Maybe we'll stick in one extra chair. But it's from 3:30 to 5:30. So you have to be there by 3:30 today, and then tomorrow. Okay, today and tomorrow. So I think because maybe you're not on the list of the students
Yeah,
in the
I know
center.
that.
Yeah. So, but the others know. Like you know about it, and the others know. So, but you can show up if you want to. And probably one chair will probably manage to get it from somewhere. Okay.
Okay.
Maybe from my office or somewhere you will get one chair and put it in. But I saw already there are 25 chairs over there. Okay, so all the chairs have been put over there. Yeah, free course, okay, or whatever. Uh, huh,
Is there like video record for that course?
I don't think so. So I don't think that room has the. I don't know unless Hondo can do. Do you have a tablet or something? You need a tablet, but Nushin has to agree to that. I can take my headset, and put it in. But she has to agree to that, right? Yeah, I don't think they thought about all that stuff, but yeah, it probably could be done, if you take a headset and, uh, if you have a tablet, you can do it, right? So, but today it might be too late. Maybe she is not prepared and all that. All right, any other questions? So I have just started grading your exams. Okay, I haven't been able to get to it because there are too many things to do. But I'll get it back to you by Friday. Okay, because this is not a big class, so for the other class, it might take a little Monday, but yours, I think I finished like maybe about 7 out of 17 or something. So I didn't spend a whole lot of time either. So, and as I pointed out, as I mentioned when I came in, when the exam was being proctored by somebody else, the problem was here. It's not with my headset. Okay, so today we should be fine. Right? So last time when we left off, we were talking about inference of networks, all right? So when you're trying to infer a network, you're going to confront three impediments in designing gene regulatory networks from experimental data. The first one is the complexity of the model. Second thing is that the data is severely limited. And then the third thing is that you lack appropriate time course data to model the dynamics. Right? So numerous approaches to network inference have been proposed in the literature, many of them based on gene expression microarray data. Right? So you see the data and then based on that you're trying to infer a network. Here we are just going to briefly outline some of the proposed methods for probabilistic Boolean networks. Okay, these are not the only kind of gene regulatory networks. There are people that study other kinds of networks, and everyone has proposed their own methods. But again, all those methods, they will suffer from these drawbacks because you have very limited amount of data. Right? So we'll just look at a couple of methods and just move on from there. Now, as first proposed, the inference of the probabilistic Boolean network is carried out using the COD, right, the coefficient of determination, right, that I talked about, and I will also discuss the main COD paper after I get done with this chapter, okay, with the details. Now, for each gene in the network, a number of high COD predictor sets are found, and these high COD predictor sets determine the evolution of the activity status of that particular gene. And furthermore, the selection probability of each predictor set for a target gene is assumed to be the ratio of the COD of that predictor set to the sum of the CODs of all the predictor sets that are used for that target gene. Right? This is how we went from Boolean networks to probabilistic Boolean networks. Right? If you remember, when you had Boolean networks, there was just one Boolean function, right? That was going to decide the update. But when you go to probabilistic Boolean networks, right, you have a number of different possible predictor sets, right, and it will update according to one predictor set with a certain probability, another one according to some other probability, and so on. Okay, and here you could assign the selection probability of each predictor set as being proportional to its COD, right? Whichever has got higher COD, that will have a higher probability associated with it. So this approach makes intuitive sense since it is reasonable to assign the selection probability of each predictor set in a PBN to be proportional to its predictive worth as quantified by the COD, or the coefficient of determination. So then there's another approach, right? A second approach to PBN construction uses mutual information clustering, right? How many people, communication people are here? A lot of you, yeah. So you know what mutual information is, you know. If you don't know, then you have to go and read, okay. Because you have to first define entropy, then mutual information and so on, okay. So these are not new methods, okay, for network inference based on genomics. It's just that the application is new, okay. And I don't have enough time to cover this method, okay. There is a paper on that. I think it was published probably in around 2000 or so, right? You can take a look. But again, it's basically the same old method for network inference, but applied to genomic data, right? So in this approach, you're going to use mutual information clustering and reversible jump Markov chain Monte Carlo predictor design. So first, mutual information minimization clustering is used to determine the number of possible parent gene sets and the input sets of gene variables corresponding to each gene. Because here you need to know which gene is going to be affected by which one, okay? Which are the parents of a particular gene, right? So they're using mutual information to figure that out. So after that, each predictor function, and then once you've figured out the parents, you need to know the function that is mapping from the parents to the child, okay? So thereafter each predict, and for that, if you want a general function, you know, you can use like I think they use a neural network, yes, because that's a function approximator, right? That's all that there is to it. So thereafter, each predictor function from the possible parent gene sets to each target gene is modeled by a simple neural network. So that's a function approximator, right? So you're going to learn the parameters. So consisting of a linear term and a nonlinear term, and a reversible jump Markov chain Monte Carlo technique is used to calculate the model order and the parameters. Okay. And again, this is not something new that is invented in the context of genomics. It is already there out there in the literature, in the inference literature. And finally, the selection probability for each predictor set is calculated using the ratio of the CODs. Now, in most expression studies, there is some degree of previous knowledge regarding the genes. There is some prior knowledge, okay, from the literature you have that play a role in the phenotypes of interest. For example, P53 in unregulated proliferation. We know that P53 is a very important gene. It's mutated in many cancers, right? So if you have prior knowledge, okay, I mean, and you know that even based on, you know, your knowledge of signal processing communications, if you have prior knowledge, all right, you want to incorporate that into your method, okay, for network inference, right? So here you're going to do exactly that. So to take advantage of this knowledge, so you don't start the inference from scratch, okay? You can, okay, you can say, but it'll take forever to learn, right? So if you want to, whatever prior information you have, assuming that it's not garbage, you want to incorporate it. That'll cut down on the amount of time that you'll have to spend, you know, coming up with a better model. So to take advantage of this knowledge and to obtain networks relating to genes of interest, it has been proposed to construct networks in the context of directed graphs by starting with a seed consisting of one or more genes believed to participate in a meaningful subnetwork. So you know that gene A, gene B, and gene C, they are related, okay? So you might seed a network, you might start it with those three genes and then grow the network, right? So given the seed, a network is grown by iteratively adjoining new genes which are sufficiently interactive with genes in the growing network in a manner which enhances subnetwork autonomy, right? So if you want to focus on a subnetwork that is, you know, controlling some behavior of the cell, right? Then you will start with some genes to seed that network and then grow from there. So the proposed algorithm has been applied using both the COD and the Boolean function influence, right? Which I didn't discuss in this class here. When we talk about the Boolean networks paper, or actually you'd have to look at the paper in order to come across the influence. This is basically, if you have a Boolean network, you come up with this network, then you want to figure out, you know, which, let's say you want to knock down some gene, okay? So which other gene should you use, okay, to knock that down? So there is a way to calculate the influence of each and every gene on that particular node, right? And that can be done using the Boolean function, the influence function for a Boolean network, right? So this measures the interaction between genes. So you could grow the seed network using the COD or any other measure of interaction, right? Whatever you like. So this algorithm has got the benefit of producing a collection of small, tightly knit, autonomous subnetworks as opposed to one massive network with a large number of genes. Such small subnetworks are more amenable to modeling and simulation studies, and when properly seeded, are more likely to capture a small set of genes that may be maintaining a specific core regulatory mechanism, right? And in fact, the next figure shows a melanoma network, right? Melanoma is a kind of skin cancer, right? And this network has been grown from 4 seed genes, WNT5A, RTN1, S100B, and SNCA. These are 4 genes that are known. The biologists know that they are important in the case of melanoma, right? So you started out with those 4 boxes like this, right? Then you can look at this box 25A, these guys, okay? And then you basically use the COD method, right, to find out which are the genes that are most closely linked with it, and you can come up with this network, right? And it gives you some insights, like some of the genes, other genes that have been picked up, they are known to be important in the context of melanoma, right, in metastatic melanoma. But some others you don't know, okay, those might represent new things. So here in this network, the COD has been used as the measure of gene interaction, and the boxes denote the seed genes, while the ellipses are the genes added by the algorithm. The solid lines represent strong connections, like you can see. The solid lines here represent strong connections. See, the COD is pretty high, whereas the dotted lines are weaker ones. And somehow I don't know what motivated them, but they said anything less than 0.3 is low. Okay, weak connection. So while the dotted lines represent weak connections, connection strengths between 0.2 and 0.3. The network that you develop by using the seed algorithm reveals some very interesting insights, which are highly consistent with prior biological knowledge derived from earlier gene expression studies using melanoma cell lines. For example, it is known that the Wnt5A gene product, that means the protein that is produced by the Wnt5A gene, has got the capability to drive aspects of cell motility and invasiveness, because the cell has to be able to move around. If the cancer is going to metastasize, the cell has to become mobile or motile. Okay, so and it is known that Wnt5A is a good predictor of that. Okay, if Wnt5A is high, if it's overexpressed, that's an indication. That's a bad sign. That's an indication that the melanoma will metastasize. Right. So that being the case, given the role of Wnt5A in metastasis, it is to be expected that genes which play a role in either mediating the extracellular matrix remodeling or interactions, such as MMP3, which is matrix metalloproteinase 3, SERPIN B2, right? That is serine or cysteine proteinase inhibitor, and MCAM. I've given the full forms also here. Okay, it's melanoma adhesion molecule or cellular movement, right? So extracellular matrix remodeling, changing the shape of the extracellular matrix, or for the cell to move, such as MYLK is myosin light polypeptide kinase, right? That will allow the cell to move, right? Would share regulatory information with Wnt5A, because Wnt5A is known to be promoting the development of metastasis. On the other hand, it is not known how Wnt5A regulation is coupled to other genes which play a part in melanoma cell proliferation, such as the MAP2 kinase 1, right? This is mitogen activated protein kinase 1. MAP kinase is mitogen activated protein kinase. Okay, what it means is we have looked at examples, and I think I discussed that when a cell is going to divide, okay, there are transmembrane proteins, okay, usually they are tyrosines, okay, that are crossing the cell membrane in the form of an alpha helix, right? And it's like something that sends a signal from outside the cell to inside. So you have this transmembrane protein. When some molecule comes and binds it outside the cell, all right, the cell division machinery inside is turned on. The thing that comes and binds the cell, that's called a mitogen, right? Mitosis, okay, is cell division, right? Mitogen is something that promotes cell division. So if that molecule comes and binds the receptor on the outside of this transmembrane protein, that will be a mitogen, okay? So that's why these pathways are called the mitogen activated protein kinase pathways, okay, or the MAP kinase pathways. So while developing this network, they found that Wnt5A also shared relationship with MAP kinase, right? And the regulation of apoptosis. These are called caspases or cysteine aspartate proteases, okay? When a cell dies through apoptosis, okay, which is programmed cell death, the cell dies a very, very neat death, okay? Everything falls apart, proteins are cut and all that, and that is brought about by these guys that are called caspases or cysteine aspartate proteases, okay? So, you have caspases appearing in this diagram, right? MAP kinase also appeared, okay? Somewhere it is there somewhere, I don't know. Yeah, see MAP kinase, you can see MAP kinase. And where is the caspase? There must be a caspase here, CASP. Or is it not there? I didn't see it there. Did you see any caspase? Oh, there it is. Yeah, caspase. And so this has to do with apoptosis. Okay, this, in fact they call it the apoptotic cascade. The caspases bring that about. One after the other, the cell just falls apart, right, and dies very, very neatly, right. The MAP2 kinase or the MAP kinase is basically when a mitogen comes and binds from outside, right, and then there is the signaling inside, phosphorylation of a lot of proteins and all that. So that's the MAP2 kinase. So here, I mean, they were not expecting that this guy and then the caspases also will have something to do with the Wnt5A, all right? But they found this, so they're saying probably there are connections over there that we do not know, right? So on the other hand, it is not known how Wnt5A regulation is coupled to other genes which play a part in melanoma cell proliferation, such as the MAP2 kinase 1, right? And in the regulation of apoptosis, such as caspase, cysteine aspartate protease 3, and BIRC1, baculoviral IAP repeat containing 1. Nevertheless, it is quite possible that high-level coordination of these activities exists through either specific circuitry or as a consequence of differing extracellular interactions which arise from metastatic cell movement. Right? When the cancer metastasizes, the cell moves. Right? So it's possible that there are other things that come into play that we don't know, we don't understand. That would reveal that kind of connection in the seed network that you have grown. Now we move on to the issue of steady state data. The problem is you do not have time course data. You don't have dynamic data. You're trying to infer a network, right? Regulatory network, right? So that's a difficult problem. So a key issue in network design arises because much of the currently available gene expression data come to us from the steady state phenotypic behavior and does not capture any temporal history, right? Consequently, the process of inferring a probabilistic Boolean network or any gene regulatory network, which is a dynamical system, from steady state data is a severely ill-posed inverse problem, right? Steady state behavior constrains the dynamical behavior of the network but doesn't determine it. There might be a lot of different systems, right, that will fit that same steady state behavior. So therefore, building a dynamical model from steady state data is a kind of overfitting, and it is for this reason that a design network should be viewed as providing a regulatory structure which is consistent with the observed steady state behavior, right? Nothing more than that, okay? Also, it is possible that several networks might emerge as candidates for explaining the same steady state data, right? So under the assumption that we are sampling from the steady state, right, all the data is coming from the steady state, a key criterion for checking the validity of a design network is that much of its steady state mass lies in the states that are observed in the sample data, because it is expected that the data states consist mostly of attractor states, right? And a number of recent papers have focused on network inference, keeping in mind that most of the data states correspond to steady state behavior. In one of these, a fully Bayesian approach has been proposed, which emphasizes network topology. And again, I mean, these papers are all there on our websites. Okay? You can go and look over there. But once again, I mean, in terms of biology, in terms of genomics, it's not that new. Okay, those network techniques are already there. You're just trying to, tweak it for that particular application. So the method that is developed in that paper computes the possible parent sets of each gene, the corresponding predictors, and the associated probabilities based on a neural network model using a reversible jump Markov chain Monte Carlo technique. So this must be the same kind of paper that we were talking about before, okay, where I said that you're going to use mutual information clustering, right? Here maybe you use different criterion, okay, but to get to the same point. And then the structure of the predictor is the same. It's like a neural network model, right? And then you'll use a reversible jump MCMC technique. And an MCMC method is employed to search the network configurations to find those with the highest Bayesian scores to construct the PBNs. Again, you want the ones that have higher scores, right? You want to use them in the network. So this method has been applied to a melanoma cell line data set. And the steady state distribution of the resulting model contains attractors which are either identical or very similar to the states that are observed in the data. And many of these attractors, they are singleton attractors. That means once you get in there, you don't get out. Okay? It's not like a cyclic attractor, right? And which mimics the biological propensity to stably occupy a given state. Furthermore, the connectivity rules for the most optimally generated networks constituting the PBN were found to be remarkably similar, with strong interactions between the components. Right? Again, I mean, this is some method they came up with, and they're trying to relate it to the real world knowledge. Now, if we consider network inference from the general perspective of an ill-posed inverse problem, then one can formalize inference by postulating criteria that constitute a solution space in which a design network must lie. For this, we propose two kinds of criteria. One kind is the constraint criteria. These are composed of restrictions on the form of the network, such as biological and complexity constraints. For example, you might say that you cannot have more than 4 levels. for going to the attractor from any state. Okay, that could be a constraint. Okay, or you can say that the attractor should be singleton attractors. Okay, you should not have large cycles, right? Even if there are cycles, maybe only 2 or 3 states. Right. Operational criteria are composed of relations that must be satisfied between the model and the data. So examples of constraint criteria would include limits on connectivity and attractor cycles. Okay, that each gene should be directly influenced only by 2 or 3 other genes. Okay, so it's a sparse connection. Now, one example of an operational criteria is some degree of concordance between sample and model's CODs, and another is the requirement that data states are attractor states in the model. All right. So you had this data, you learned this network, right? Now you go and analyze that network, construct the attractors. Those data states, those attractor states better belong to the same data. Okay, I mean overlap with the same data. The inverse problem may still be ill-posed with such criteria, but all solutions in the resulting space can be considered satisfactory relative to the requirements imposed by the criteria. Right? And this kind of approach has been implemented by finding constituent Boolean networks satisfying constraints such as limited attractor structure, right? Transient time, how many steps you take to get to the attractor, and connectivity, and so on. Okay, these papers are already there. They are published. They are there on the website. It's, I think, 2005 paper, right? So if you want to take a look, you can. If you want to present one of these in class, that's fine too. Right? All right, any questions so far? Now, in addition to the ongoing effort to infer probabilistic Boolean networks, there has also been a continuing effort to infer Bayesian networks and dynamic Bayesian networks. Right? What is a Bayesian network? A Bayesian network is essentially a compact graphical representation of a joint probability distribution. Right? So it tells you, like each gene is affected by its parents. Okay? So you have a conditional probability transition table. It will tell you that if the parents are taking these values, then the child will assume this value with probability so much. Okay? That kind of information you have. So the representation takes the form of a directed acyclic graph in which the nodes of the graph represent random variables and the directed edges or lag thereof represent conditional dependencies or independencies. Right? That's what I was saying. If the parents take value, one parent takes 1, the other one 0, then the child will take certain value with certain probability. Right? So the network also includes conditional probability distributions for each of the random variables. Now, in the case of genetic networks, the values of the nodes can correspond to gene expression levels or other measurable events, including external conditions. Like, let's say you put in a drug, right? That could also be a conditioning variable, right? Now, there is a precisely characterized relationship between certain dynamic Bayesian networks and probabilistic Boolean networks in the sense that they can represent the same joint distribution over their corresponding variables, right? And in this sense, PBNs are more specific in the sense that the mapping between PBNs and DBNs is many to one. So that a DBN, a dynamic Bayesian network, does not specify one single PBN, a unique PBN. Okay, several PBNs might be mapping to the same dynamic Bayesian network. But again, I mean, these connections also have been worked out, and there are papers that are available that try to nail down the connections between dynamic Bayesian networks and probabilistic Boolean networks. All right, any questions? Yes, sir. Go ahead.
My question is, do they use as a model? I mean, here different kind of like dynamic Bayesian network, probability Bayesian network, things like that. I mean, do they really use them in designing drug, or it's just like for the matter of?
See, okay, yeah, okay. In theory, they can be used for designing drugs, okay, but remember, right? In practice, the problem is that, you know, if you come up with a probabilistic Boolean network, the dynamic Bayesian network, maybe it's used. The Bayesian network might be used, okay, there are more people working. The probabilistic Boolean network, you have to first validate it using experimental data, okay. If you validate it, then you can use it for designing drugs. I will talk about that, okay, how to intervene, because, you know, if the network is not behaving in a desirable way, you want to intervene, okay. Which gene should you intervene with? Things like that. There are ideas that we will throw out there, okay, but I'm not aware that too many what should I say, drug designs have been done based on that, simply because, you know, nobody has taken the time to go and collect data from the real world and validate these networks, all right? Yeah, another way in which you can see, because these are very sparsely connected networks, all right? So what you could do, and I think that has a better shot at succeeding, is instead of trying to infer a network from scratch just by using data, take the pathway knowledge. Biologists have marginal information about interactions between different molecules. Okay, they call that pathway knowledge. If you go and look up under biological pathways on the internet, you'll see there are tons of them. Okay, so you might want to take that information, right, and then update that using, like whatever other time domain data that you have, right, and then you've actually put in some structure into the model, right? Because the PBN, everything is up in the air. Okay, you're going to update according to certain probabilities. What are those probabilities going to be for selecting the predictors? Okay, we don't know, right? So I don't think that this has been completely done. Okay, I mean, even for Boolean networks, I don't think that kind of modeling actually has been done in terms of linking it up with the real world. Okay, these are just ideas that have been thrown out and worked upon, and the theory has been developed, but I don't think anybody has developed a drug just based on the Boolean network and all that stuff. I mean, we are trying to do some of that here, but not necessarily with a Boolean network, right? Maybe with a Bayesian network, conditional probability tables, you can probably do something, right? Okay. See, it's again, I mean, if there's a rich theory of stochastic processes, okay, and let's say the cell phones are going to make use of that, okay, but until you get the cell phones to work, all right, nobody cares how rich your theory of stochastic processes is, okay, whether you're using the dominated convergence theorem, whatever, all the, measure theory, right, nobody really cares, all right. That's good theory, right? So here also, I think until you take it to the finish line, the full impact will not be realized, right? And maybe it's up to some of you guys to do that, okay? Okay, the next topic is intervention, all right? And in intervention, what you're trying to do is, let's say, you have a gene regulatory network that got messed up because of some mutations, okay? So now you want to intervene, okay? If it's going to lead to cancer, you want to try to see, okay, which is the best gene to intervene with, okay? Or even some of you are working on agri-life projects. Okay, if it's a plant, water use problem that you're looking at. Okay, you want to make the plant more water efficient. Okay, drought tolerant. Okay, so you might have some signaling pathway. You might build up some regulatory network, and you want to be able to predict. Okay, which are the genes that I should alter so that the plant uses less water. Okay, that's an important problem. Okay, in plant biology also. So those kind of things we are going to discuss in this chapter on intervention. Okay, but before that, I want to spend some time. I want to spend like a couple of lectures, maybe today and Friday, possibly even Monday, talking about the COD. All right, I'll discuss the COD paper. All right, and also the explanatory material that goes with that paper, right? This intervention thing I can cover quite quickly. It's not a problem, okay? Because and this is something that I have myself have also worked on that, okay? So now, so let's switch gears now and go to the. So I think any questions? So we are on page 629 over here, okay? So another 70 pages. 70 pages and we'll get done, okay? But I want to first now go to the material on on the COD paper, the explanatory material on the COD paper, right? And I think wait a minute, okay, there should have been, yeah, COD explain, yeah. So first I'm going to cover my explanatory notes, okay, on this stuff, and some of this stuff will be quite familiar to you because I know there are many people here with background in communications and in signal processing and all that, and these are not new techniques, okay? So, if you already know this stuff, this is just like a review, but I'll go through that because these ideas are used in the COD paper, okay? So this is about, so the COD or the coefficient of determination, that's a technique that we are using basically to do expression prediction. Like if you have a microarray, all right, I mean, what is my best estimate for the expression level of a gene given no data? Okay, that's what I want to see first. And then I want to see what is the best estimate if I have some other data, okay, if I have some other predictors, all right? And the amount by which the estimate improves, that is basically the COD, okay? That's what I defined earlier, right? And that's what we'll see in this paper. Now, in order to talk about the COD, we need to understand a few concepts, all right? So the paper that we will discuss is the general nonlinear framework for the analysis of gene interaction via multivariate expression arrays, all right? And here I'm trying to fill in some of the details, okay? So this is for the book that we were working on, myself and Dr. Doherty and this is like a draft of that chapter, or an initial draft when I was preparing some of this material. So, but I think, and that semester, that was in the summer of 2004. So I was teaching a course, basically this course only for the first time. Okay, writing the notes and all that. So let's fill in some of the details. Okay, because if you don't know the details, then it'll be difficult to follow that particular paper. Right. First one. Given a real valued random variable y, the best constant predictor of y, right, in the absence of any observation, right, in the absence of any more side information, is the mean of y. Okay. So did you all know this, or if you didn't know, we are doing the proof here, because remember I had to teach this also to undergrads. Okay, they didn't know, and some of them were not happy because 303 was not listed as a prerequisite. So if I offer an undergrad version of this course later, I will list 303. That's random, I think, random signals and systems. Yeah. So I will make that a prerequisite because, I mean, in previous year, huh?
That's junior course, but this 489, oh, this
480, it's the number is 419. It has become a regular course. I offered it a number of times as a 489, and then it became a regular course. But this time I didn't club it with 669 because I didn't know how to handle this problem. If they don't have 303 again, they'll come, and the biology part is fine, but once it gets into the stochastic stuff, then it has happened once that some people got really upset. Okay, in most cases they were happy because they're learning so much of new material, right, that they are willing to stretch and endure the growing pains. Okay, even learning some of the stochastic stuff, because I covered that stuff, tried to explain, but one or two people, they were a little bit turned off. So maybe next time I have to list 303 as a prerequisite for this class, because these kind of things would be covered in 303. So the idea is, so you have a constant predictor C, right? So the you look at the mean square error. So you take the difference between the random variable and the predictor, right? Square that, take the expectation. That's the mean square error. So you do the expansion, then you get this expression. Now you're going to take the derivative, set that equal to 0, right? To minimize this error. So you differentiate with respect to C, and setting the derivative to 0, right, you're going to get C is the mean of the random variable y. Okay, so that's the best constant predictor. And to make sure that in fact this thing is minimized, when you choose C equal to the mean of y, right, you take the second derivative, and the second derivative is positive, so you do have a minimum. Okay, so that's the first thing, first fact, right? Now the second fact is that if you have data, because remember when you're doing the COD, you want to get the best predictor, right, in the absence of any data, in the absence of any side information kind of. Then you want to also get the best predictor with the data, right? And then you want to see by how much you improved. Okay, that's what the COD measures. So fact 2, if you have data, the minimum mean square error estimate of a random variable y given an observed random variable x is the conditional mean or the conditional expectation of y given x. All right. So did everybody know that, or most people knew that, right? I mean, if you don't make any distributional assumptions, right, and your criteria is the minimum mean square error, the best estimate is the conditional expectation, right? Right? Because you are conditioning this on the data. Okay, over there you don't have any data, so it is just the expectation. So again, if you want to do a proof of that, right? So the proof of that is like some completion of squares. Okay, I mean it's one of those problems. I don't know. Is it easy to do the regular proof? I mean the proof that I'm doing is a little bit of cheating because it's like I know the answer and I'm going to plug it in and try to manipulate and show that that is the answer, right? At least that was the proof that was shown to me when I was a grad student. Okay, so I don't know. Has anybody seen a better proof? Right? There must be, because somebody must have guessed that. Okay, I mean, right? I mean, because you're students in communications and these areas, right? I mean, so you're experts in this, right? But anyway, I'll do the proof that is good enough to explain to undergrads. So you're looking at the mean square error. So y hat is the estimate of y. Okay, so you take the square of this, take the expectation. That's the mean square error. Then then you know the answer. Okay, the answer is the conditional expectation of y given x. Okay, so I'm going to cheat a little bit. I stick in this answer here, minus expected value of y condition on x plus expected value of y condition on x. Didn't change anything. Now I'm going to expand. Okay, so this term and then this term. So it's like a plus b squared. So I'll have these three terms. Okay, the first square term is here. Then this is the last square term, expected value of y condition on x minus y square of that. And then you'll have the cross term. That is the product of this guy, and then the expectation of that. Okay. Agree? Everything is fine. Okay. Now we are going to focus attention on the last term here. Okay, and we'll show that that is 0. How do you do that? Well, instead of taking the expectation over all the variables, okay, I'm going to do it in 2 steps. I'm going to condition the expectation on x, all right, and then take the overall expectation, all right. So whatever I have in here, all right, this thing, I can write it with another expectation outside and then everything conditioned on x. That's what I did here. Okay. Now, if something is a function of x, all right, and you condition that on x, you don't take any expectation, right? I mean, right? Because you're not averaging it out over x. So y hat is the estimate of y. Okay, that is the function of the data. Okay, function of x. Similarly, the expected value of y conditioned on x, that's also a function of x because you condition the expectation on x, right? X is not averaged out. So that means when I take this conditional expectation here, this thing, right, y hat minus the expected value of y conditioned on x, it's like a constant. It can come out of this expectation. So it came out because you're conditioning the inner expectation on x, right? You just took that out, okay? Because that's like a constant. You can take it out, and then you are basically left with this expectation of this quantity, okay? Now here, when you try to evaluate this, okay, look. Again, I mean, this expectation is conditioned on x. So you already have expected value of y conditioned on x. If you take another expectation condition on x, it will not change anything. So it will still stay expected value of y conditioned on x minus, all right, now this is y, expected value of y conditioned on x, right? So this is 0, right? So, so that being the case, all right, this being equal to 0, this last term is going to drop out, right? If the last term drops out, notice that this thing does not depend on the data, right? It's expected value of y conditioned on x minus y squared. Doesn't depend on data. The only thing that you have freedom in choosing is this y hat, right? That is your estimate. So, and this is a squared quantity, right? So it is non-negative. So to minimize this, you should choose y hat equal to expected value of y conditioned on x. Okay, that's what we did. So note that the second term does not depend on y hat, and the first term is non-negative. Thus, expected value of y hat minus y square is minimized if we choose y hat equal to the expected value of y conditioned on x. Right? So in the presence of observation, the best minimum mean square error estimate is the conditional expectation. The only problem with the conditional expectation is that if you want to calculate it, right, you'll need to get the conditional density function, right, and then you'll have to calculate that, okay, and that can be a mess, okay. So you want to see if you can get to the answer without having to calculate that. So because of that, you're not going to try to calculate the best estimator, right? Minimum mean square error estimate. Maybe I will confine myself to the classes of linear estimators, okay? I will not get the overall best, but it will be a lot easier to calculate, right? If it is the best one, I need the conditional density function, right? So it's a lot of information that is needed, okay? So instead of working with the conditional expectation, I may look at the best linear mean square error estimate, okay, of the random variable y given the observed random variables x1, x2 up to xm, right? You have observed these guys, you want to make an estimate of y, okay. So for that, the estimate is going to be a linear function, okay. It's going to be a linear function of these variables that you observe. Linear or actually affine because you're adding a 1 times a hat, okay, and then there are these parameters that you're going to determine. So again, this is similar to whatever we were doing earlier. Remember we talked about the Bayesian, the Bayes error being the minimum, okay, right? I mean the Bayes classifier that being the best one, but I mean it's too hard to learn that, okay? So you put in some structure. In the process, you have given up the overall best, okay? You've sacrificed a little bit, but you've made life a lot easier, okay? So here we want to approximate it like this, okay? We want to find out a hat that'll give me my best estimate for y hat, okay, based on the observations that I've made. And for this, you can get a formula, not very difficult. So again, the mean square error is the expected value of y minus y hat square. So it's y minus VA, okay? V is that vector containing the x1, xi's, and then 1. And then A, and then you just do the computation. This is y minus VA transpose times y minus VA. You expand this out, you get it something like this, right? Then you'll do exactly what you did before, right? Like take the derivative, right, with respect to A, right? Now you're taking a derivative with respect to a vector, okay, vector of parameters, and you set the derivative to 0. Then you obtain this equation because for this one there's no A, so you'll get 0 here. So 2A transpose R minus 2C transpose equals 0. Then you write this as RA equal to C, or A hat, the best estimate is R plus C, all right? Where what is R? R is basically the autocorrelation of V. So it's V transpose V, expected value of V transpose V, and C is the cross correlation between Y and V, right? So that's the expected value of Y and V, right? So I think I must have defined that on the last slide, okay? It's given by this, all right? Did we not define it? We should have. Yeah. See where A hat is R plus C, all right? And then R is the autocorrelation, C is the cross correlation, right? And R plus represents the pseudo inverse of R, right? So you can do a proof of this. So since y hat is a linear function of these guys, it will have to be of the form y hat equals VA for some vector A in Rm plus 1, right? And now I take the difference between y and this VA, and I take the derivative, set that equal to 0, which is what is done on the next page, and then the solution comes up here, okay? Now one more thing that we want to nail down is what is called the orthogonality principle. Right? Are you familiar with that or no? Yeah, the error is orthogonal to the observation. Okay, that is going to be needed. Okay, so from 1 and 2, okay, from these two relationships, we see that the minimum mean square error, which is expected value of y minus y hat, instead of y hat you'll put v a hat, is going to be expected value of y square minus c transpose a hat, right? I mean, because when this relationship is satisfied, 2a transpose r is 2c transpose, you put in 2c transpose, you put in c transpose a here minus 2c transpose a, so you got 1 minus c transpose a hat, okay? Then stick your expression in for a hat, okay? And you will get, this is the expected value of y square minus expected value of c transpose. Well, c was the cross correlation, so you'll get yv times a hat, okay? So you can write it out like this, expected value of, and then you can factor out the y, you write down y minus v a hat, okay? I'm trying to get to the orthogonality principle from here, okay, from this optimal solution. So now also, you can check that expected value of y minus va hat square will be basically this multiplied by itself. So you'll write down y times y minus va hat times y minus va hat. You multiply this out, you get y times y minus va hat minus, you can write this as a hat transpose, and again you can check the math, okay? If there are any problems, you let me know, okay? But you can write it out like this, all right? Now comparing these two guys, 3 and 4, all right? You can see the same term is appearing here and here, right? The first term is the same, which means this guy must be equal to 0, right? So this fellow, this second term must be 0, which you can write it out like this. And this is the orthogonality principle, because it's telling you that the error is orthogonal to the observation, okay, when you're doing this kind of linear estimation, right? For the best, and in fact, I think the orthogonality principle is used for deriving all these Wiener filters and all those things, all right? So, okay, you must have seen them somewhere, or look back in your notes, okay? Now, my intention is that I need to use this orthogonality principle to derive some of the equations in that paper, okay, that we are going to discuss next time. So let's try to derive equation 5 in that paper, right? And I can walk you through the steps. So in that paper, we are interested in determining the best linear mean square estimate of y given the random variables x1 through xn, right? So then in this case, A will be looking like this, B, A1, A2 up to AN, and the linear estimate will be of this form, y hat equals VA hat, right? Where V will be having 1, x1 up to xn, exactly the theory that I developed, okay? And then multiplied by this vector, okay? So you can write it out as B hat plus sigma I going from 1 to n, AI hat xi. Now without any loss of generality, we are going to assume, and as it is assumed in the paper, that we are going to assume that the xi's have got 0 means. Okay, if they don't, that can always be corrected by this term here. Right, there's a b hat term here. So now from 3 in the derivation of fact 3, right? See this one. See, we found an expression for the minimum error, right? See 3, right? The minimum error, if you're going to use the optimal linear predictor, is given by this. Okay, so we write that expression out, because remember, to calculate the COD, I need to get the error without any observations for the best predictor, okay? Error for the best predictor in the presence of observations, and then I take the difference and the ratio, and I will get my COD, okay? So here we are trying to get closed form expressions, okay, for the COD, right? So from 3 in the derivation of fact 3, you will get this expression epsilon opt is this, okay, which is expected value of y square minus expected value of yv times a hat. So that's expected value of y square minus expected value of y times v a hat. Okay, v is this, 1 x1 up to xn, and a hat is this, all right. So you multiply this out, you get expected value of y square minus it'll be b hat times the expected value of y times 1, so mu y. And then for the others, all right, it is going to be basically the expected value of y multiplied by each of these xi's. So you get a1 hat multiplied by y x1, expected value of that, and so on. Okay. So now I introduce the correlation coefficient between xi and y. Right. I'm going to write things in terms of the correlation coefficient. Right. So the correlation coefficient between xi and y is given by this formula. You take the mean from the random variable, mean from the other random variable, multiply it out, take the expectation, divide that by the standard deviation of each random variable. Right. That's the formula for the correlation coefficient. Right. Now since the mean for the random variables that we are using, the mean is 0, you can write down that the expected value of xi, so these terms will drop out, this and this will drop out, and expected value of xiy is going to be the correlation coefficient multiplied by the standard deviation multiplied by the standard deviation of the other one. Okay, so I can stick this in over here, right? I can stick this in here, and so and even for this I can write down in terms of the mean and the standard deviation, right? I mean, because you know that, right? I mean, when you calculate the variance, you can express this in terms of mean and the standard deviation. It will be like mu square plus sigma square, right? So now sigma square y is expected value of y square minus mu y square. Wait, maybe I skipped something. Let me see. I don't want to skip anything, so let's see. So substituting b into a, right? You get this relationship, all right? So this is for the error of the best linear predictor without any observations, okay? I get this relationship, and furthermore, you also get that let's see. Okay, so this is without any observations. This is the expression that you got for the error, okay? Now, if you make observations, okay? No, wait. No, that is with observation. Epsilon opt is with observations, right? I mean, because you have the xi's that are being observed, okay? So that is with observation. If you don't have any observation, we said that the best predictor is the mean, okay? So what is the error of the best predictor, the mean square error? That is the variance, right? I mean, okay? So you now have expressions for both of these, right? So the COD is basically the error of the best predictor without observations, right? Minus the best predictor with observations divided by the error of the best predictor without observation. So you put in sigma square y, here you'll get sigma square y, and then you subtract out all of this stuff, right? But then you know that expected value of y squared you can write in terms of sigma and in terms of the mean, right? So you can simplify this and you get this expression, right? So you just simplify this and you finally get this expression here, okay, which is the expression that is given in equation 5. This is a closed form expression for calculating the COD. Right, in the case where you're using a linear predictor. So that's for linear prediction, a closed form analytical expression for the COD can be obtained, right? Okay, I think let's stop here for today, right? I just have a little bit left and then I will go on to the discussion of the actual paper next time. Maybe I stopped a couple of minutes early, but that's okay. It's raining and, it's a mess.